Hey 201 Lab, this is the AV lecture for Lab 25. In this AV lecture, we're going to be talking about taste and smell. Before we get into specifically taste and smell, let's just generally talk about senses. On a daily basis, we're constantly being bombarded by stimuli, and stimuli are chemical or physical components in the environment that elicit some sort of response when our body receives it. And in order for humans and other animals to make sense of their environment, we have evolved specialized receptors which receive specific stimuli and turn that into electrical signals that our brain can process and turn into our senses like sight, sound, taste, and smell. Now, these specialized receptors are called mechanoreceptors, photoreceptors, chemoreceptors, and proprioceptors. Mechanoreceptors are going to be specific to detecting mechanical stimuli, and we're going to have these receptors in our ear. Photoreceptors are going to detect light, and we find this in our eye. In the posterior portion of our eye, we have a structure called the retina, and the retina receives light that enters our eye. Chemoreceptors detect changes in the chemical environment around us. And this is what we're going to be discussing today, taste and smell. And proprioceptors is our body's ability to know where it is in space. So to illustrate this, if you ask me to close my eyes and then ask me to touch my nose, I would be able to do that without seeing either my hands or my nose. And that's because of our proprioceptors. Now, receptors are, you can think of them as extensions of nerve endings. And again, they respond to specific types of stimuli. And when receptors receive some sort of stimuli, whether it's a chemical, some sort of mechanical stimuli, or a photon of light, these receptors convert that stimuli into an electrical signal that's sent to the brain, and only in the brain is it perceived as taste, smell, sight, or hearing. Now let's get into taste here. As we all know, we have 5 primary tastes: salty, sweet, sour, bitter. This one may seem new to you, umami. And this is kind of this earthy, meaty taste. And this is actually when you are stimulating your umami senses, you are actually detecting amino acids in the food. Now, when we went over the brain, we discussed cranial nerves and how they were involved in taste. And cranial nerves are we're going to have 3 cranial nerves that are involved in taste. The glossopharyngeal nerve is going to detect taste or chemical stimuli in the posterior third of your tongue. The facial nerve is going to detect taste in the anterior 2/3 of your tongue. And the vagus nerve is going to be detecting taste throughout your tongue. This diagram is representing your tongue, and these colored areas are representing spaces on your tongue where you are relatively more sensitive to a certain taste. So on the back of your tongue, you're more sensitive to bitter. On the tip of your tongue, you're more sensitive to sweet. This does not suggest that in only these areas can you taste bitter, sour, salty, or sweet. That's not what we're representing here. This just represents areas of relatively high sensitivity. And the reasons for this become clear if we put this in an evolutionary perspective. On the back of your tongue, this is going to be an area that will potentially stimulate a gag reflex. And in nature, poisonous foods are typically bitter. And if an early human were to eat a poisonous berry, that poisonous berry would pass over the back of the tongue, stimulating the gag reflex to cough that poisonous berry up. Now on the tip of your tongue, you are relatively more sensitive to sweet tastes. And that's likely for a similar reason. In nature, sweet foods are limited both in time and space. But sweet foods are going to be high in energy, they're going to be high in calories and sugar. And so it's going to be a really valuable food source that early humans wouldn't have come across that often. Now we can go to the vending machine and get a Snickers bar anytime we want. But in early evolutionary time for humans, sweet things were highly limited. And we wanted to know if we were eating something sweet. So often, if you think about a newborn baby, what do they do when they pick things up? Well, they stick their tongue out and put whatever object they have in their hand right on the tip of their tongue. This could potentially be a way to identify sweet things that would be high in calories and high in energy. In lab, you will discuss more hypotheses as to why you are sensitive to these tastes in these areas on the tongue. Now let's get into specifically how taste works. In anatomy and physiology, we don't like the word taste. We have a better word for that, and that's called gustation. This is the anatomical term for taste. And the process of gustation begins with putting food in your mouth. And you're going to chew this food, and that's going to help break down the food. But also, you're going to start producing saliva, and saliva is going to break that food down. And chemicals in the food are going to get mixed into the saliva, and they're going to wash over taste buds, which are found on the tongue, and they're the bumps, or they're found within the bumps on your tongue. And the taste buds are going to receive the chemicals that are mixed in with the saliva, and those are our chemoreceptors. And those chemoreceptors, when they become stimulated, will send a action potential or electrical signal to the brain, and it's only in the brain do we process that information into something meaningful like taste. A critical component for gustation is for the food to break down in saliva. Saliva is mostly made up of water. It's 98% water, but it's also going to contain mucus, electrolytes, immune cells, and enzymes. And this is going to serve to break down the food and release the chemicals and get them into the saliva. If you do not have saliva, you will be unable to taste. That's how important this is to the process of gustation. Once we have stimulation of the chemoreceptors at the taste buds, we are going to send an electrical signal to the brain, and first it's going to go to the medulla oblongata. The signals are going to be relayed to the hypothalamus and the amygdala, and then to the thalamus. And recall that the thalamus is going to be kind of the switchboard for your brain. It's going to receive a lot of sensory information, and it's going to redirect that information to the proper location for processing. And so the thalamus is going to send the signal to the postcentral gyrus, and this is where we become conscious of taste. Everything before that was either only chemicals or electrical signals. It's only when we get to the postcentral gyrus do we become conscious of taste. Now there's also going to be another pathway to the orbital frontal cortex shown here. And this is where we're going to integrate the taste with sight and sound. And at the end of this AV lecture, we will go over why it's important to pair senses together and integrate them in the brain. So that was a broad overview of the physiology of taste. And now let's talk about the specific histology of gustation. Earlier in the AV lecture, I mentioned that on your tongue you had these bumps, these raised projections that contained your chemoreceptors. And if we zoom in on an area on your tongue, and we took a cross section of that area, you would see an image similar to this. And these raised projections are called papillae. And within each papillae contain taste buds. And we've seen papillae before. When we went over the dermis, we had papillae in the papillary layer of the dermis, and those were raised projections of the dermis into the epidermis. And these appear to be similar. The papillae on your tongue, we have these raised projections here. Now, we discussed how saliva was important to gustation and that it's actually extremely necessary. And that's because as you break down the food, the chemicals become dissolved in saliva. And they actually wash over the papillae, and it gets pooled up in these ridges between papillae, in these spaces between papillae. And right on the sides of the papillae, we're going to have our taste buds. So let's zoom in to that area. Just for orientation, we're looking at a papillae here, and this is one taste bud. There's another taste bud here, another one here, and another one here. And there's also one over here. And each one of these taste buds is a chemoreceptor. Now, the saliva, as we're chewing our food and the saliva is breaking the food down, that saliva is going to wash over the papillae, and it's going to pool up in the spaces between the papillae here. And that gives the taste buds the opportunity to pull chemicals from that saliva. And as they pull those chemicals from the saliva, the taste bud becomes stimulated. And that's when we have action potentials sent to the brain via the pathway we just discussed. And we have perception of taste at the postcentral gyrus. So taste buds are contained within these papillae here. We have different types of papillae, fungiform, folate, and circumvallate. We are showing folate papillae here. And taste buds are going to, again, be our chemoreceptors. And they're going to be bundled in clusters of about 80 cells. So we're packing in a lot of different cells, a lot of different chemoreceptors into that taste bud. Now we're going to wrap up gustation with some interesting facts here. If you've ever heard the term, it's an acquired taste, this is a real thing that simply means that as you get older, your taste buds become less sensitive to particular tastes. And so if you remember growing up, you may not have liked foods like tomatoes, broccoli, Brussels sprouts, fish, coffee. And that's because those foods had a really strong taste. And your taste buds were young, they were eager, they were highly stimulated by these strong flavors. And that probably turned you off to those foods. But over time, your taste buds, just by aging in general, but also by damage, those chemoreceptors become less sensitive to those sorts of flavors. And you actually learn to like those tastes. And that's simply because your taste buds are just not as sensitive. And so that's what it means to have an acquired taste. You have almost 10,000 taste buds inside your mouth. The number of taste buds actually differs by sex. And if we think about this under an evolutionary framework again, females would have the responsibility of rearing children. And because of that, there was a lot of pressure, natural selection pressure, to identify foods that were poisonous and identify foods that were nutritious, because a female was not only looking after herself, but also if she was pregnant, she was looking out for her baby. And so it's hypothesized that females developed more taste buds and became more sensitive to taste relative to males so that they could protect their pregnancy. And finally, taste is the weakest of our five senses. And now we're going to move into a different sense, but one that is highly linked to gustation. Second part of this AV lecture is about olfaction, which is smell. Now, just to get oriented, we're looking at a cross section here, a mid-sagittal section of the skull. We can see our brain here. This would be our oral cavity. So this is our maxilla bone. This is our frontal bone. This is our ethmoid bone. And this is our nasal cavity. This image here is just a blown up image of this right here. Now, smell is very similar to gustation in that it still involves chemoreceptors. And the way that smell is going to work is, first, odor molecules are going to be around the environment at all times. Chemicals are going to be floating around. And as they float around, we call these volatile molecules because they're floating in the environment. They're going to enter our nasal cavity through our nostrils. And the odor molecules, the chemicals, are going to dissolve in the olfactory mucosa, which is a structure in your nose that's mostly made up of mucus. And as that dissolving of those chemicals takes place, your chemoreceptors are going to receive those chemicals. That is going to initiate an action potential, and it's going to send an action potential via the olfactory nerve, which is shown right here. Remember, this is our cranial nerve number 1. Now, that action potential is going to be sent to the temporal lobe. That's going to be the primary pathway. And it's at the temporal lobe where our brain perceives that electrical signal as some sort of smell. Now, there's also going to be a secondary pathway. One is going to be to the hippocampus and to the amygdala. This is the limbic system. This is where we're going to form memories of certain smells. And we're also going to have an emotional response linked to a certain smell. And to illustrate this, think about your favorite meal that your grandmother or your mom or your dad, grandfather cooks. Maybe, or maybe even the smell of cookies. That likely, when you smell that smell, that will not only make you think about the cookies, but it will also make you think about your grandmother, your grandfather, your parents, whatever that smell is linked to in your memory, that will also be recalled. And then the amygdala, that will elicit some response, some emotional response too. So if you always remember what your parents' home smells like, when you go home and you get those chemicals again, that's going to elicit some sort of emotional response about being home. Now, we also have a secondary pathway to the hypothalamus, and this is where we're going to have hunger stimulation or hunger perception. And this makes a lot of sense. If we think about when you smell dinner cooking, that is going to make you hungry. And that's all because of this secondary pathway to the hypothalamus. Now, these examples of the secondary pathway were very modern, but if we think about this under an evolutionary framework, like we've been doing the rest of this AV lecture, these secondary pathways become to make I would argue make more sense. First, the hippocampus, this is again going to be memory formation. This is a way for early humans to link what is a good smell and what is a bad smell. What is the smell of an enemy, something that might want to eat us or something that might want to hurt us, and what's the smell of food. And also this hypothalamus, this secondary pathway to the hypothalamus, this is going to be a way for early humans to pursue a particular food that might be edible to them. And so if they smell, for example, a certain plant that's edible, or they smell an animal that is prey for them, this will stimulate the hunger center and it will encourage those humans, those early humans, to pursue that prey. Olfaction is a sense that adapts to the environment relatively quickly. If you've ever come home and your roommate has forgot to take the trash out and your whole apartment smells like trash, you will adapt to that smell relatively quickly. And even without removing the bag of trash, you will just become used to that smell in your apartment. Now, this is important because olfaction, as well as our other senses, are critical in detecting changes in the environment. And this becomes important because if our bodies can detect changes in the environment, we can then respond to the environment. If we only responded to whatever the majority of the stimuli are in the environment, we would never be able to detect changes in the environment. And those changes in the environment can potentially become harmful to us. And so with smell and with these other senses, what's really important is being able to detect changes in environmental conditions. Just to conclude, these are some tips for your next practical quiz. For gustation, you should feel comfortable describing the pathway of gustation, what are the nerves involved in gustation, and where is gustation processed in the brain. You should feel comfortable identifying structures associated with taste using histology, so the papillae, the taste buds. You should be able to tell us why saliva is important for gustation. What does it do? Why is it necessary? You should also feel comfortable describing the different hypotheses that we presented in this AV lecture as to why you may be more sensitive to certain tastes in certain areas on your tongue. As far as olfaction, you should feel comfortable identifying the structures associated with olfaction. Those include the olfactory nerve, or cranial nerve 1, the chemoreceptor at the olfactory mucosa. You should be comfortable with the primary pathway of olfaction as well as the secondary pathways of olfaction. You should also be able to give us hypotheses as to why those secondary pathways of olfaction evolved.